Students, welcome to Microbiology Lab. So today's experiment is gram staining. So gram staining is also called as Gram's method. So it's a method of staining which is used to distinguish or classify the bacteria into two large groups called as gram positive and gram negative. Okay, so this technique was introduced by a Danish bacteriologist called Hans Christian Grams, and that's why the name Grams staining has come. So here the gram positive bacteria stains as dark purple color and gram negative bacteria will appear as pink color. So this is a widely used staining method in microbiology and clinical microbiology labs as well. So this is the first step in identification of bacteria. And there are four major basic steps for the gram staining. First is the application of the primary stain called the crystal violet to the heat fixed smear of bacterial culture. Then you have the second step called the iodine application, where the iodine will be applied to the slide and the iodine will be combining with the crystal violet and trap it inside the cell. Then we have the third step, that is the decolorization with 70% ethanol, and the last is the fourth step, that is the last step, which is the counter staining with safranin dye. So here we use two stains, that is the primary stain crystal violet and the counter stain saffron. So let's see what are the requirements needed for the experiment. You need the crystal violet dye, then the grams iodine, 70% ethanol and safranin, and water, distilled water, and then you need the main thing called the slide, that is a micro slides. You have the bacterial culture here. I have taken the curd and now then loop and also the dropper for applying the primary stain on the slides. Okay, so and also you need lamp facility because we are here we use the smear. Okay, heat fixed smear is used for the experiment. So we have to apply the smear bacterial culture on the slide and then we will heat fix it. Okay, that is by using the lamp. We have lab facility here, so we'll be heat fixing the smear. The first step is the heat fixing of the bacterial smear. For that, you have to take a loopful of bacterial culture and you have to make a smear. Okay, the smear should be made. And then you have to fix it by passing the slide over the flame. Okay, now the smear is fixed now. We have heat fixed it. So allow it to cool for some time. Okay, so I already told you the basic step starts with the application of the crystal violet dye. So we have the crystal violet dye. So after cooling the bacterial smear, we will be applying the primary stain, the crystal violet. Okay, that is, we'll be applying it over. We'll cover the smear with this crystal violet dye for 1 minute. The time should be noted, it's for 1 minute. And what I hope the slide has cooled up. So what we do is we'll cover the smear with crystal violet dye. So a 1 minute of crystal violet dye should stand over the smear. So once it's done, we have to wash it off with a tap water or distilled water. So after the 1 minute is over, we have to wash it with tap water. So the 1 minute is over now. We'll wash it off with tap water by you have to make sure that you don't wash off the smear and you have to apply the water very gently over the smear so that the violet color will be washed out. Okay, now we have washed the smear with water. The next step is the application of iodine solution. So this is the grams iodine. Now we'll apply the grams iodine over the smear. Okay, that is also we have to make it for 1 minute. Cover the smear with iodine solution and wait for 1 minute. Okay, now the 1 minute is over. We'll wash it off with the same water again. So the third step is the alcohol decolorization by alcohol, that is 70% ethanol is here. Here the alcohol will be taken and the slide should be washed off with alcohol. Until the violet color runs off, wash it with ethyl alcohol. Okay, now the next step is the application of the counter stain, that is the safranin. So once it is washed off, wash it with clear water again. Okay. Now we have to take our safranin and cover the smear again with the safranin dye. Wait for 1 minute. After 1 minute, you can wash it off and then you can view it under the microscope to see the stained organisms. So the 1 minute is over now. Wash it off with the clear water. Okay, try the slide. Keep the slide for drying. Just after the slide is dried, you can view it under the microscope. So this is how the dried gram stain appears, the dried smear after the staining. So now the step is you will be viewing it under the microscope to distinguish whether it's a gram positive or a gram negative. So we have the microscope here. I've already told you discussed you with the different parts of the microscope. So now we'll focus it.